In this lesson, we look at synaptic transmission, and here are the objectives. As a reminder, the signal that is traveling within a neuron, the action potential, is an electrical signal, but the signal that travels between neurons from one to the next is chemical, and now we're going to look at how that happens. The entire junction between one neuron and the next is called the synapse, and the space between them is called the synaptic cleft. The neuron leading into the synapse is called the presynaptic neuron, and the neuron leading out of the synapse is called the postsynaptic neuron. So in this diagram, we're looking at a signal that's traveling down from the top. The top neuron is the presynaptic neuron. So the action potential, that depolarization, travels down the axon and reaches the end of the axon, called the axon terminal, and then it depolarizes the axon terminal. Now, waiting in the axon terminal, there are vesicles filled with neurotransmitter. That's what I'm drawing here. These are vesicles filled with neurotransmitter. And with depolarization of the axon terminal, voltage-gated calcium channels open. So this is meant to be a side view of a voltage-gated calcium channel. And then calcium flows into the presynaptic neuron. We've seen voltage-gated sodium and potassium channels in the axon when we were talking about the action potential. Now we've got voltage-gated calcium channels. And calcium serves as a signal for these vesicles to fuse with the membrane and for exocytosis to release the neurotransmitter into the synaptic cleft. So this is showing exocytosis, and then the neurotransmitter gets released into the synaptic cleft. Once it's there, it can bind with postsynaptic transmembrane receptors. That's what I'm drawing here. And those receptors can be of different types. If they are, for example, a ligand-gated ion channel, then they can let in ions. For example, sodium. And that would be the case if this neurotransmitter were acetylcholine, which we write as capital A, capital C, lowercase h. That's acetylcholine. And this were a nicotinic acetylcholine receptor. The nicotinic acetylcholine receptor is on the postsynaptic neuron, and when it binds acetylcholine, it opens a cation channel and lets sodium in. And it's called the nicotinic acetylcholine receptor because it binds to nicotine and responds to it as well. Now, that influx of sodium is going to cause a bump in the membrane potential of the postsynaptic neuron. So if the y-axis here is membrane potential and the x-axis is time, it's going to cause a positive bump, a depolarization in the postsynaptic neuron's membrane potential. And if there's more neurotransmitter, we would see a bigger bump. That makes this a graded potential, and because it's positive, it's depolarizing and it's bringing the neuron closer to its threshold potential, we would call that an excitatory graded potential, or an excitatory postsynaptic graded potential. Now, there can be other postsynaptic transmembrane receptors. For example, I'll draw one over here that would respond to a different neurotransmitter, say the neurotransmitter GABA, that's spelled like that, and if it bonded to the neurotransmitter GABA, it could open a chloride channel. Now, chloride is a negative ion, and therefore that's going to hyperpolarize the postsynaptic neuron, make it more negative, and bring it farther away from its threshold potential. And once again, if more of that GABA neurotransmitter were released and more of these chloride ion channels were to open, then we'd see a more negative graded potential, bringing the neuron further and further away from its threshold potential, making it less and less likely to fire an action potential. And this postsynaptic neuron is receiving multiple inputs like this, not just from this one presynaptic neuron, but from many, many presynaptic neurons. And therefore, it's seeing the combined total effects of all of these ion movements in. And therefore, a combination of multiple excitatory and inhibitory postsynaptic graded potentials. And it's by summing those, the total effect of those brings this neuron either closer or farther away from its threshold potential, makes it more or less likely to fire an action potential. And it's through that summation of all of these different graded potentials that this one neuron can integrate inputs from multiple neurons that are synapsing on it. We should also look at how neurotransmitter is removed from the synaptic cleft and its action terminated, especially because this is an important mechanism of action for many drugs. Now, neurotransmitter, once it's in the synaptic cleft after it's been released from the presynaptic neuron and after it's bound to the postsynaptic neuron transmembrane receptors, can just diffuse away, but it can also be taken back up into the presynaptic neuron, and we call that reuptake. And then it can be repackaged into vesicles and re-released later. There are a variety of drugs that can interfere with that reuptake process by blocking the transporters that do that. An important class of drugs in this category are the selective serotonin reuptake inhibitors, also called the SSRIs. Serotonin is a neurotransmitter that is important in regulating mood and sleep and digestion, and the SSRIs block its reuptake into the presynaptic neuron, and by doing that, they prolong its persistence in the synaptic cleft and therefore alter transmission in synapses that use serotonin. And they're used very prominently in the treatment of mood disorders and anxiety. Another drug that blocks neurotransmitter reuptake is cocaine, but cocaine does so in a much less selective way. It not only blocks the reuptake of serotonin, but it also blocks the reuptake of norepinephrine, which is a neurotransmitter involved in regulating alertness and in preparing our body for fight or flight, and it also blocks the reuptake of dopamine, a neurotransmitter that is central to our brain's reward system. And it's the combined effects of all of those persisting longer in the synaptic cleft that lead to cocaine's effects on the brain and body and its potential for abuse. In addition to reuptake into the presynaptic neuron, another way that neurotransmitter can be removed from the synaptic cleft is by being broken down by enzymes. And in fact, this is the way that acetylcholine is removed from the synaptic cleft. It's broken down by an enzyme called acetylcholine esterase. Now, if you inhibit the enzyme that breaks down acetylcholine by using a drug called an acetylcholine esterase inhibitor, then the acetylcholine is no longer being broken down and it's going to persist in the synaptic cleft longer and transmission is going to be altered. And so acetylcholine esterase inhibitors are used in the treatment of dementia to try and increase the transmission through neurons in the brain that use acetylcholine as their neurotransmitter. So that's a look at synaptic transmission as well as some of the ways that it can be altered by drugs.